The MAPK-RAS-RTK pathway is a crucial signaling cascade regulating cell growth, cell differentiation, and cell survival. The receptors, which are responsible for the signal transduction in mitogen-activated protein kinase pathway, are receptor tyrosine kinases. There are a variety of ligands involved in this pathway, mainly growth factors such as epidermal growth factors, platelet-derived growth factors and fibroblast growth factors, stimulating cells to undergo cell division, cell differentiation, and cell survival. The pathway begins when a mitogen binds to an RTK at the ligand-binding domain on the extracellular side. Binding of the ligand leads to conformation change in the receptor, leading to dimerization of two RTKs. This dimerization brings the intracellular kinase domains of the receptors into close proximity, which leads to their autophosphorylation on specific tyrosine residues on the cytosolic side of the receptor. The tyrosine kinase domain is activated, creating phosphotyrosine docking sites for various intracellular signaling proteins that relay the signal. To these phosphotyrosine docking sites, an adapter protein, called GRB2 docks. GRB2 has an SH2 domain which binds to these phosphotyrosine residues. One or both of the GRB2 SH3 domains bind to a SOS protein through its proline-rich region. SOS is actually a guanosine nucleotide exchange factor, helping in GTP exchange. SOS has a RAS-GEF domain which is responsible for activating RAS by facilitating the exchange of GDP for GTP on RAS. The RAS is now activated and dissociates. It moves along the membrane to interact with a kinase known as RAF. RAS is a molecular switch. It has intrinsic GTP-ASE activity, and its activity triggered by activated RTKs is short-lived. The RAS gaps induce the activated RAS to inactive itself by hydrolyzing its bound GTP to GDP. RAS is no longer active and it does not interact with RAF. The pathway is stopped. Considering the active form of RAS, after binding with RAF, also known as MAP kinase, the RAF is recruited to the cell membrane, where RAF undergoes a conformational change and becomes activated. Once RAF is active, it phosphorylates and activates the next kinase in the cascade, MAP kinase, commonly referred to as MEK. MEK, in turn, phosphorylates and activates MAP kinase, also known as ERK. Activated ERK either phosphorylates proteins or enters the nucleus, where it interacts and phosphorylates one or more components of the transcription regulatory complex. This will change gene expression and cause cellular response such as cell division or survival. Receptor tyrosine kinases, RTKs, have wide-ranging applications due to their central role in cell signaling, growth, and differentiation. RTK levels and mutations can serve as biomarkers in diagnosing cancers and predicting prognosis. RTKs like FGFR and EGFR promote tissue repair and cell growth, making them valuable in regenerative medicine. RTKs such as VEGFR play essential roles in blood vessel formation, angiogenesis. Drugs that target VEGFR are used to manage diseases that involve blood vessel growth. Please like, share and subscribe and ring the bell icon for more videos.